done over the past summer and into the fall with Professor Susan Cushman. We've been looking at the effect of round gobies on the benthic lake macroinvertebrate community in the Finger Lakes region. So, as I'm sure many of you know, the round goby is an invasive species that has begun to invade the United States. It originates from the Ponto-Caspian region, and it was first discovered in the St. Clair River in 1990 in the Great Lakes region. And it's slowly been migrating down to the Finger Lakes region in New York State. It's a recent invader of many of the Finger Lakes within this region. So Oneida, Onondaga, and Cayuga Lakes, there are established populations. And we think that it's been coming through these water bodies through the Erie Canal. So a little background on the round goby. It's a benthic generalist feeder, so it feeds on pretty much anything it comes across. And it also is a very successful invasive species. So it's temperature tolerant. It has an optimal temperature of about 26 degrees Celsius, but has a wide range throughout the lake. It also is temperature dependent. Its spawning is temperature dependent, so when it gets to the right temperature, they'll start to spawn. They also eat macroinvertebrates. That's their main source of prey. And we know from previous studies that look at stomach contents and feeding trials that they have preferred prey species that they like to eat. What's also interesting is that many prey items of the round goby are also invasive Ponto-Caspian species. So that's drifted mussels and recent invasive amphipods coming into the area. So, I'm sure many of you also know that lake macroinvertebrates are very important to the community. They're an important food source for many of the fish in the ecosystem. So they're really important, and we wanted to look at differences. And there's been previous studies, specifically by McKenzie in 2006, looking at how these communities change once an invasive species enters the ecosystem. So if you look at the right hand, or the right column slides, you can see that these communities are impacted when an invasive species is in the ecosystem, such as the round goby. You can also see from this image that certain species, such as true flies, worms, snails, are impacted strongly. And also you can see that there is an invasion factor, especially of the descended mussels, like the quagga and zebra mussels. So knowing this and knowing that the round goby has prey preferences, we know that they have a stable food source in the ecosystem, ranging from native macroinvertebrates to invasive macroinvertebrates. And this aids in their egg production and survival rates, again making them a really successful invasive species. We also know that they have preferences, so they feed on soft-bodied and hard-bodied prey items. Which again makes them a generalist feeder, but also there will be differences in prey abundances. So this study, we have a little bit of background knowledge going into it. We know that invasive species can impact macroinvertebrate communities. So we wanted to keep in mind the taxonomic richness and look at the taxonomic richness in these communities to see if that was being impacted by the presence of the round goby. We also know that the round goby has prey preferences, so we wanted to look at specific species that the round goby was feeding on to see what their abundances were like based on pre-invasion to post-invasion. So there's also been studies, like I said, showing that if a round goby is presented with a hard-bodied prey items such as a mussel or a soft-bodied prey item such as an amphipod, they're more likely to choose the soft-bodied prey. So knowing this, we had a study done in 2012 by the Watershed Assessment Association, and what they did in 2012 was take an overall benthic survey of the Finger Lakes. So they looked at all the Finger Lakes, took benthic samples, water quality, and data like that. Which gives us a really great baseline because the round goby was not in Cayuga Lake, or was not known to be in Cayuga Lake before 2012. So what we wanted to do was look at Cayuga Lake compared to a control such as Seneca Lake, which does not yet have a population population of brown goby and compare taxonomic richness and prey species since this lake. So we hypothesized that there was going to be an impact on both taxonomic richness and prey, and specific prey items that we know the brown goby prefers. And we predicted as well that there was going to be no change in Seneca Lake just because there's no populations of brown goby that we know of. So for our experimental design, again, we were basing this off the WA sampling methods because we wanted to keep things consistent and have a good comparison. So we looked at the sites that they went to in 2012, and we set this up as a backy design. So we have our before, after, control, and impacted. Before invasion, 2012. After invasion, 2019, this past summer. Our control was Seneca, which again has no population of brown goby, and our impacted, which is Cayuga Lake. The way we chose sites was we looked at the macroinvertebrate orders that were documented in 2012 and chose sites that had at least 10 macroinvertebrate orders. So we knew going to these sites that there was supposed to be a large range of species. And then we chose 7 sites on Seneca and 8 on Cayuga, mostly in the north to mid range of both of these lakes. So for our field sampling, we sampled this summer, so through the months of July and September, which again is consistent with the 2012 study by the WAA. We sampled at 1 meter depths, which is again what the study was done in 2012. And our sampling methods consisted of using a conar grab sampler, which was 23 by 23 centimeters. And we would send this down, get our benthic sample. We would sieve through this sample and then place it in an Nalgene bottle and preserve it with 95% ethanol. We also, at these sites, took water quality data and data such as plant abundance and dominant substrate at each site. So this past fall, in the lab, I sorted through all of these bottles following the New York DEC protocol from 2018, and also went through these samples in the same way that was done in 2012. So also too, to keep things consistent, we attempted to get 300 individual counts. So we subsampled through our bottles until we got at least close to 300 individuals. And these individuals were identified through order and family, and anything we had left, if there was anything left from the sample, we would re-preserve in 70% ethanol. So for our data analysis, again, we looked at taxonomic richness between Cayuga and Seneca Lake and 2012 to 2019. So we used an ANOVA with that. We also looked at preferred prey items, and we looked at percent abundances from 2012 to 2019. For our abiotic data, we also used an ANOVA, and we focused on water temperature and dissolved oxygen at these sites. And then for our substrate and plant abundance, we looked at percent present in both lakes and in both years to see if there's been any change. So for our results, we found that there's a statistical difference in Cayuga Lake from 2012 to 2019. So there has been a decrease in taxonomic richness in Cayuga Lake, which could be due to the brown goby being present in this lake. We did not find any statistical difference in Seneca Lake. So we also look, if you remember the table from the previous, a slide towards the beginning, we wanted to see if certain species have changed in this ecosystem since 2012. So we looked at the species that were present in 2012 and compared them to our list from 2019 to see what individuals were missing from our samples. And so as you can see, we had various mayflies, caddisflies, dragonflies, also various beetle species, snail species, worms, and fingernail clam. So a pretty wide range of individuals were not in our 2019 sample as they were in 2012. So then we broke down our prey preferences into hardbody and softbody. So our hardbody was quagga and zebra mussels, the invasive species, and we found that there's been a significant increase in these populations since 2012, which makes sense. They're a highly successful invasive organism, and they have done really well in the Finger Lakes region. There's also been previous studies shown that while the goby heavily predates on these individuals, or on these populations, they reproduce at such a fast rate that it's not really being impacted. And then we looked at our soft-body prey, so amphipods and chironomids, and we found that there are significant decreases in both lakes, so Seneca and Cayuga, since 2012, which could be due to varying factors, environmental conditions, or even competition from invasive amphipods stemming from the Ponto-Caspian region as well. And then for our chironomids, we didn't really find any statistical results. Okay, so again, we looked at temperature and dissolved oxygen at these sites, which was done in 2012 as well. And we found that there has been a significant decrease in water temperature in both Seneca, in both Seneca and Cayuga Lake since 2012, which could just depend on the year or on an overall change in the community. Also, too, it's interesting to keep in mind the optimal temperature of the goby, which is 26 degrees Celsius. However, they're temperature tolerant and have a wide range in their ecosystem. Also, we looked at dissolved oxygen and found a statistical result in Seneca Lake. Dissolved oxygen has increased in this lake since 2012. And then we looked at dominant substrate, which is that top table there, and we found that there's been changes since 2012. We found that in 2019, there's a complete dominance of cobble and gravel substrate in Seneca Lake, which is slightly concerning just due to the fact that we know that round goby prefers a harder substrate, and that gives it the potential of being very successful in Seneca Lake. We also found that in Cayuga Lake, there's been a shift towards more silt substrate, which again can be due to environmental factors. And also the round goby is still very successful in this ecosystem. So then we also looked at plant abundance, and that is varied throughout 2019 and 12. So what this means, this study gives us a really good baseline of what changed in Cayuga Lake and what we can predict to change in Seneca Lake, because we know that the round goby will eventually invade this lake ecosystem. So we have a better sense of what will change in the lake, and we also have a better sense of how these changes can impact desired game fish or threatened native species that we want in the lake. Also, too, we know, we were reaffirmed that the round goby invasive presence will alter macroinvertebrate communities throughout this ecosystem. So that ranges from preferred prey items, such as the ones I talked about, and also to the Ponto-Caspian species that this species natively preys on, and now, because they have also invaded this ecosystem, they're preying on as well. And also, in the larger scheme of things, if we're looking at the ecosystem functions and trophic cascades and things like that, we can predict that there'll be changes not only to the round goby, but also because there's been water quality changes, and those are just things to keep in mind, especially when we know kind of the biological functions of these invasive species. And this can help inform us on invasive monitoring programs in the future. So with that, I'd like to acknowledge the Hobart William Smith Biology Department and the Provost's Office, the Finger Lakes Institute for the donation of their vote this summer and their time, my research team, Kate Foley and Ali Seminar, and Dr. Susan Cushman. I'd also like to thank the AFS for their travel award this year.